and start. So again, hi everyone, hope you are fine. My name is Karina Ladeira and I am the Young Investigator Representative of the International Comet Assay Working Group, that is a specialist group of the European Environmental Mutagenesis and Genomics Society. As you can know, this is the second and last Meet the Expert of this year of 2022. And this initiative has the aim of dynamize the dialogue between mainly young researchers who use or have the interest to use the Comet assay and the experts that use already from few years and many years the Comet assay. So I really hope you enjoy this panel of experts that we choose for this section, this second meeting. I have some information before we start. Again, the meeting will be recorded and shared and streamed on YouTube, so you can revisit as much as you want. You can explore also our website and see another videos that we have, more technical, and also the first meeting experts that was organized in February this year. And you can still drop questions in Zoom chat, in YouTube, and also to raise your hand and directly ask the experts in the end of each presentation. I will do my best also with Vanessa Andrade and Anya Averick that are co-hosting this webinar with me to check all your message. And now let's start with our first expert. That is Dr. Gunnar BRUNBORG from Norwegian Institute of Public Health in Oslo, Norway. And Dr. Gunnar, he studied physics at the University of Trondheim, specializing in biophysics. And after some years of research in radiobiology, including a year in Oxford and at the Medical Research Council in London, he specialized in toxicology at University of Oslo. Besides other activities, he followed by more than 30 years at the Norwegian Institute of Public Health as a researcher and later as chief scientist and department director. And he retired in 2015, but fortunately for us, he's still very active as a scientist in collaborative work in Norway and abroad. And is a very special expert to us because he like a lot and have a personal interest in the alkaline elution. And his ambition has been to employ basic physics and chemistry to understand and improve the Comet assay. So Dr. Gunnar, it's a pleasure to us to have you here. And I would like you to share your screen and give us your presentation about electrophoresis and Comet assay, an essential stage which is often misunderstood. Thank you so much for having you here.
Thank you, and I hope I hope that I'll be giving you some useful information. I hope that some of you are fresh to the Comet assay because I'll start with some basics actually. Allow me to cough a bit, sorry. Can you see my screen now?
Yes.
This is actually the view from my home office this morning. What's happening now? Can you see another screen? Oh, that's it. Okay, no. Sorry. Yeah, anyway, there's a bit of snow in Oslo this morning, so you can see what it looked like when I looked out of my window this morning. Okay?
Okay.
So I am going to talk about electrophoresis in the Comet assay. And the electrophoresis is defined. You go to Wikipedia and you'll see how it is defined. It's actually the movement of a charged particle in an electric field. It's very well described in Wikipedia. And since DNA is negatively charged, it moves in this electric field. When DNA is in a viscous fluid or in a matrix, the movement is a bit delayed or restricted. Can you see? Do you see the complete screen, or do you also see our images? Do you see the whole screen now?
We see the whole screen, yes.
Okay, that's great. Fantastic. Okay, anyway, the total movement of DNA in this matrix depends on the electric field strength. And it depends on the duration of the treatment and also of the viscosity of the matrix. If you have water, for example, then everything would flow everywhere because the viscosity of water is so low. But if in this case, in our case, the DNA is in agarose, then the movement is really quite inhibited. So, however, please note that there is nothing here about the current, the electric current. But even so, the electrophoresis conditions were in the past often described as 300 milliamps, 25 volts, and with an approximate liquid volume. And it was said that it should last for 20 to 30 minutes. So then I had to start with some basic physics here. And the basic physics is done in this fantastic drawing, which I also made. And it shows an electric circuit. It shows an R, which is a resistance, and PS, which is a power supply. And it shows U, which is a voltage, and I, which is actually a current, and showing the direction of the flow of the current. And the Ohm's law describes the relationship between the voltage and the resistance and the current. And I hope you all remember this from primary school or secondary school, I'm not sure. So U equals R times Y. That's essential. So what happens then if you have an electrophoresis tank as an electric circuit, which it actually is? Then the electric parameters are defined by its total resistance, or the inverse, which is the conductance, which are determined by sidewell dimensions, platform dimensions, the volume of the liquid, specific conductance of the liquid, and the electrode conductance, the material and dimensions of the electrodes. And also then, of course, the power supply. And the power supply can be an electronic power supply, or it could also be a battery. It has a voltage, but it also has an inner resistance, which I might mention a bit later in my talk. Okay. So then this is an example. Here we have parameters in the Comet assay being defined as a voltage of 25 volts and a current of 300 milliamps, which is the measure often cited in protocols for the Comet assay. Then the equation is 25 equals R multiplied by 0.3 amps, which is 300 milliamps. And that gives an R of 83.3 ohm. As a consequence, then the resistance of the system is defined using the Ohm's law. If you then alter, and this is important, if you then alter some of the resistance parameters and the volume, and at the same time you require the voltage and the current not to be altered, then you violate Ohm's law, which is not good. You eat the cake and you try to keep it too. So you can't do that. So if you want to change the current, then you are actually at the same time you're altering the resistance of the system. But do you alter then the electric field properties so that you also alter the electrophoresis? No, you don't. So this is quite good. What is happening in the Comet assay electrophoresis? You have nucleids, which are structured packages of DNA. Also, there is a very nice description of that in Wikipedia. And they have a total negative charge, so they move in the direction of the the positive electrode. And these nucleids, they move in agarose when subjected to this electric field. In the Comet assay, we electrophorese many of those structures in a tank and on a platform. The aim is therefore to create an electric field on the platform which does not vary in any direction for nucleids placed on the platform. So how do we do that? It's actually quite simple. We use this two-dimensional structure with long electrodes on each side of the tank, which they determine the y-axis, and the field between them should then be fairly constant, and the x and the y-axis, okay? And then you may think, well, what's the role of the current? I'm very, very happy when I prepared this talk, I was very happy because there was an incoming question concerning the current, or one of the participants. Hopefully this person is listening to me now, because it really gave me the opportunity to try to describe why the current is not important at all. So you may think of the volume in the electrophoresis tank as layers in the z-axis. That's the depth of the liquid of the tank, is that of moving ions. They move then in the x-axis. The structures to be electrophoresed are placed in one such layer, and adding more layers below or above by adding or removing the volume will not change the electric conditions of the structures. This is quite logical, actually. So then I have made, ages ago, I made some sort of a spreadsheet which allows you to try to analyze the electric conditions in electrophoresis tank. It's a spreadsheet that you put in your data, or your tank dimensions, for example, and the conductance of the liquid, and also the voltage, and etc., etc. So then you can do calculations. And then in this case, I used my tank, and I put in a volume of 1.44 liters. And then, as you can see with A, B, C, D, that calculates actually the voltage, the voltage field, both in the side wells, these are the A and D, but also on the platform, which is B and C. And the platform voltage field strength would then be a bit below 1 volt per centimeter. It says 0.94 here. So that's quite useful if you want to analyze it. And then what happens then if you add more liquid here? If you add, for example, instead of 1.44 liters, you add 1.6 liters. Then you can see, actually to the right here, you see how the voltage increases on the platform. You can see here, but that's not the voltage strength. This is simply the voltage relative to one of the electrodes. But here at the top, you can see simply a graphic presentation of the liquid, which is the red line here. The top of the liquid is the red line. Anyway, here you see you added much more liquid, 1.6 liters instead of 1.44 liters, and then you ended up with hardly any difference in the voltage. It's now the voltage field strength is now 0.925. It was 0.934, I think, in the previous picture. So as a consequence, then you can actually make a model which demonstrates that there is hardly any effect of adding more liquid to the field strength on the platform. There is a tiny effect which I might discuss a bit later, which we have time to. Then this analytic system is actually quite useful also, because if you put in too little buffer here, if you put in 1.1 liter simply in this rather large tank, then you get a warning. It says too little buffer. So the spreadsheet might be of help, but it's quite simplistic, and it's really not correct, because I'm not good enough in analyzing the real distribution of the electric fields in these volumes. I don't know how to do it, and I was never able to teach myself how to do it. So then you should also measure the electric field, and most people do nowadays. Actually, when they do Comet assay, they measure the electric field. How do you do that? You use a standard digital voltmeter. You use normal probes of the voltmeter. It costs 20 euro or something, 30 euros altogether. So it's very, very cheap. You can also get very, very expensive ones, but you only need a cheap one to do this. You add your volume of electrophoresis liquid to the tank. Then you switch on the power supply and you adjust its output voltage. Don't limit the current output. That's actually an error which some people do. They set the current of the power supply to a limited level, and that you shouldn't do that. Then you place the probes. It says probles, but it should be probes on each side of the platform, or you use a defined distance between them. Then you record the reading. Then you calculate the electric field by dividing the reading in volts by the distance between the probes. in centimeters, then you obtain the value voltage per centimeter. And that is actually the driving force in the Comet assay. But there are some points to remember when you do this, and also in red it's suggested how to solve them. You must not use a very low volume. What happens then? The liquid layer above the slides may vary locally because it's almost impossible to level the tank completely. And the accuracy of the liquid volume becomes critical. So use a large volume. But beware, increasing the volume has secondary effects. What are they? More current leads to more heat dissipation in the tank during the run. The temperature is not critical, but should be kept constant. The temperature could be anything between 4 degrees and 15 degrees. Probably you wouldn't find much difference in electrophoresis. But keep it constant during the run, please. But what you can do to reduce any heating, you can circulate the liquid. You can take it out on one side using a tube, and then have some sort of a coil immersed in ice water on outside the tank, and then pump things through. I have described this in one of my papers, and it gives more stable conditions. More current gives higher voltage drop along cables and electrodes. This is often forgotten. So if you increase your volume, then there is more current coming out of the power supply, and there is a higher voltage drop along the cables. So you should use short cables. You can even use platinum. You should use platinum electrodes, being as thick as you can afford. But what you should and could do, which is very, very simple, you could simply measure. You can touch the both ends of the electrodes with these probes, the voltmeter probes, and then you can measure and see whether there is any difference in the voltage you measure between them, and you touch electrodes themselves at the side closest to you, or on the other side more distant to you. And you shouldn't find too much difference there. I mean, I would hope that you don't find more than half a volt. So if you find 25 volt or possibly 24 volts between the electrodes closest to you, then you should find, well, let's say half a voltage on the other side. But you shouldn't confuse this with the actual driving force on the electrophoresis platform, because what you measure here is simply the voltage in the metal of the electrodes. Okay. And check the voltage before and after adding more liquid. That's important, because all power supplies, they cannot produce infinite levels of current. They go up to a certain level, and then a certain amount, let's say 4 volts or something, and then they start leveling off because they're in internal resistance within the power supply or in the battery. So check the voltage before and after adding more liquid. And it might happen that your power supply doesn't like that much liquid, and then you have to reduce the liquid, or you have to get another power supply. But do turn up the voltage to the level you want it to be. Okay, so the expected implications of the electrophoresis theory are that the Comet tails would increase linearly with the electric field on the platform. That is U, isn't it? And the Comet tails increase linearly with the duration of the process. Actually, it's not U, it's actually U divided by the per centimeter, isn't it? And the third factor would be the strength of the matrix, that is agarose. And if you have a very strong and concentrated agarose, then the movement of the charged particles would be slower, and the tails formed in the Comet assay would be smaller. So then that factor is inverse, isn't it? Whereas the voltage per centimeter and the time duration is linearly affects the percent tail DNA. So this has been tested experimentally. There is a paper made by including me, but the first author is Amaya. She is probably listening, isn't she? And then what we did here, we simply systematically measured the the effect of unwinding time, electrophoresis conditions, etc. The unwinding time is not relevant for this talk, but the aggregate concentration is definitely so. And then you could actually see that it's so that if you electrophoresis for 40 minutes rather than 20 minutes, then you get twice as much DNA damage being observed. And if you electrophoresis at 2 volts per centimeter instead of 1 volt per centimeter, then you also get twice as much apparent DNA damage. However, and there is a big however here. At first, but well, before saying however, I should say that you can actually use these results if you want to compare experiments between different labs or different experiments in your own lab, if you have varied the electrophoresis conditions. Then this should allow better comparison of different sets of data. This I find very, very important, actually. We are constantly attempting to compare our results, and we are doing large human biomonitoring studies. And if you want to compare these data, then I think one should take into account or take into reality the possibility of doing these corrections. Now comes the however. The ring tests have shown large interlaboratory variations in the Comet test results, even when using standardized conditions. How is that? And we have been struggling with this for ages, actually. There's been a number of ring tests being done to identify the differences and the cause. Different labs do get different results, very large differences. Why? Well, the ring tests that have been tried, they imply circulating cell samples for independent Comet analysis, circulating slides for independent scoring, all to identify the possible causes of variations, and comparing results using defined treatments. The gold standard here, if you want to have a defined treatment, then you should use ionizing radiation. Ionizing radiation of low energy transfer, that means low LET, as we say, produces well-defined effects on DNA at known rates per DNA unit. It's for human cells, it's very, very close to 1,000 single-strand breaks and outer labile sites per cell. So, including samples irradiated with known doses could be used as reference. However, there are still very large differences between labs per gray. And I'll show you here an example of this. We observed this, and it was published with Andrew Collins as the first author in 2008. And this shows the dose response curves. That means tail DNA damage on the ordinate axis, and on the x-axis is the X-ray dose. And you can see that the X-ray dose is the same, I think, in all curves, isn't it? But the ordinate axis vary a lot. So there is actually substantial differences here. In particular, the one right on the bottom right is very, very low. It only goes up to less than 30%, whereas in another curve, like the one here to the bottom left, that goes up to 70% or something. So it's actually a lot of difference. And that came as a surprise to us, and we didn't like it. How about all the other factors? Are they also important, such as the lysis, the duration, pH, denaturation, scoring, different cell types, cells, different radiation qualities when you do this with ionizing radiation, or different radiation doses or post-radiation conditions, repair of single-strand breaks? It's very rapid. And if there is unexpected or unknown repair after radiation before you do the Comet assay, then you might find that the damage is largely reduced. So then we designed a ring test, but this is history now. And I find it a bit awkward, but also I'm very, very happy that we have now published these results. We wanted to compare radiation units in different laboratories, and to do that, we used dosimetry systems. So we measured the doses in each of these labs, and then we tried to find the correct, let's say, correct dose in each lab. And then we used human peripheral blood mononuclear cells And we wanted to, well, the question was, do lymphocytes vary in their response to ionizing radiation? The Comet assay experimental protocols which were used were in the ring test protocol, were either the favorite protocol of the lab or a standard protocol specified for the ring test. So irradiations were then carried out of cells in suspension or cells already embedded in agarose. So that was to control for what I just talked about, that was the post-irradiation repair. I mean, if you irradiate in embedded cells embedded in agarose, so then you irradiate your slides or your films, and then you can dump it into lysis solution a few seconds after radiation. But if you actually have cells in suspension, then you must dilute them with agarose and mold the layers on the glass slide or something, and then that takes time. And also the cells are heated up when you mix them with the molten agarose. Then also some cells were sent to Oslo for additional scoring. I don't mean cells, I mean samples actually. So what we wanted to study whether there is significance of different scorings, and then we analyzed all data statistically. The embarrassing thing is that we had our first discussions in 2004 and 5, and the protocol was made. My first version was in 2009, and we did our experiments and we reported them in 2010 and 11. And we had our first data analysis in 2014, and the first draft was there in 2017. The first submission was in July 2022, and now it's accepted for publication in Mutation Research. I'm very happy to tell you this, and if this presentation had been this spring, let's say, then I don't think I would have dared to talk about this at all, because it's been hanging over me for so many years that I should complete this. But it's been a nightmare, actually. And I would, first of all, I would recommend people planning such a thing that they have very standardized reporting schemes. You should actually, people should put the data right into a database. That would save a lot of work and also reduce the possibility of errors. I'm afraid it's not available on PubMed yet, but it is available as a PhD file. If you're desperately interested, then please do contact me and I can send it to you. But it will be available soon. Anyway, so what did we do then? This is a table showing the parameters that we used. Have I used too much time already? I think so.
Yes, I will ask you just to rush a little bit.
Yeah, I will speed up now, but it's not so much left actually. This shows the electrophoresis conditions. I don't have to talk about them. They're more consistent for the standard method being described than they are for the chosen or the lab specific protocol. Okay, and here this is a very important result. Here we show how much the dose can vary between labs. Now I'm talking about the radiation dose. There is actually 46% difference when comparing the highest and the lowest value. So when you believe that you are irradiated with a special radiation dose, then you might be completely wrong, and you cannot compare it with the result from other data unless you have calibrated your radiation source. Here you see data showing dose response curves. That means on the alternate axis there is a tail percent DNA under different conditions, and each color here is one lab. To the right, you see what happened. To the left, you see the nominal doses without correction for radiation doses, and to the right, you see what happens to the radiation doses if you correct for radiation doses. And you can say that there is much less variation between the labs if you are using the standard protocol, for example. Embedded cells, that's the very best result, isn't it? And then you have alanine corrected dose. The alanine corrected dose are actually the real, so to speak, doses here. And then there are more results. Differences were less when irradiating cells embedded in agarose rather than in suspension, that you already seen. That is because we reduced then the post-radiation DNA repair. And we also found not much result from repeated scoring, so I'm not going to talk about that at all. And we also tried preparing mononuclear cells from 8 donors in my lab. And that gave actually indistinguishable dose response curves. So the correction, the last point here is actually that correction for electrophoresis conditions did lead to reduced variations. And how we did that is actually a bit tricky. It's not so easy. Firstly, we had to find the correction factors for each lab, which is shown in this table. For the L1, L2, etc. are the labs, and then the LS is the lab-specific protocol, and the S is the standard protocol. Here you have all these correction factors, and they were used. And then we found that there were indeed effects of correcting for these protocols. This is, if you have time, then you can read this legend more carefully, and then you will see, you understand what it means. Anyway, you can see that there are variations in the spreading of the data between the labs here. Then we did a linear mixed model analysis, statistical analysis of these data, which you can see here. That's one of my last slides. And this is quite important because the linear mixed model here shows that if you correct, and the complete correction is termed ABC, that is correction for both the time and the voltage per centimeter and the agarose concentration, which is here. So the ABC is all of it. And the ordinate axis here shows the percent of the variation which is statistically explained by the laboratory itself, and whereas so by correcting you reduce the error represented by varying the electrophoresis conditions. Okay, and you can see that the ABC is lower than the blue point here. The blue point is before correction. And you can see that this, particularly with a lab-specific protocol, then there is a distinct effect of the of correcting for the electrophoresis conditions. So this is actually then ultimate proof from the ring test, showing that you should correct for the electrophoresis conditions, and the electrophoresis conditions are actually deterministic for the output in the Comet assay. So I think that is it, isn't it? Yeah, this is by my daughter, and she's pregnant, and I'll probably be a grandfather in 2 weeks or something, and that's me, and this is my sailing boat. So that's it. I think I'm done.
Thank you so much, Gunnar, and congratulations for being a granddad.
Thank you.
So as you already referred, there was some questions that were posted in the Google Forms, and
yeah, that's right.
I will start for doing that questions. I already put one here in the chat in order to everybody can read also. So the question is, are the 300 milliamperes recommended in almost all protocols mandatory? Or is it possible to increase the buffer level over the slides to maintain temperature and voltage uniformity across the platform, but with a higher current?
I think anyone listening to me now should probably be able to answer that question. And the answer is definitely do increase the current. Don't leave it at 300 milliamps. If you have a normal tank and you have 300 milliamps, then you are left to make that work with a defined voltage. Then you are left with a layer of liquid above the slides, which is so thin that you can't trust it. So please increase it. You might try to double the volume, for example, and if you have, but it depends on the power of your power supply. It must be able to give much more than 300 milliamps. So that's the first question. That's my response to that. And if this person who submitted the question wants to get more information from he or she can simply contact me, no problem.
Okay, thank you so much. We have another one. What do you think about flash Comet assay version electrophoresis stage?
I think it's a nice method. I would welcome any modification of the Comet assay, and this is how we do research. We try different methods. I don't think it has a lot to offer. I mean, it cuts down the electrophoresis time a lot. However, to save a coffee break of 15-20 minutes might not be so important. So being able to carry out the electrophoresis during 2 minutes instead of 15-20 minutes might not be so essential. And it's claimed in the paper that it's more sensitive than the standard assay, and that I simply don't believe. And I don't think that the demonstration I've seen of that doesn't really convince me. And in this paper showing this or claiming this, actually they don't go down to the levels of exposure that we have actually worked with. We have found that we can demonstrate down to 0.2 grays or something. So anyway, but it's a nice method, and the reason for being able to do this is that you have completely different ionic properties of the liquid, electrophoresis liquid, when you use the flash Comet, and that's the reason why you can do it so quickly.
Okay, thank you, Gunnar. We have another one. Can DNA damage be damaged by the electrical current used in electrophoresis?
I would say no to that. There is so little current, and the forces are so small that there would be no possibility for the DNA to be pulled apart or something, or broken or something in the Comet assay.
Okay. Now we have a big question, because the person who put it had an example. So I think I put here. So there are some doubts even watching the electrophoresis video on the ICAWG YouTube page, and people weren't sure how to calculate the electrophoresis runtime. So in the video, there is an explanation on how to adjust runtime, which changes according to the size of the tank platform, and that each laboratory must measure this initially, and then measure the voltage in the tank with a voltmeter, divide the value on the other, and calculate to arrive the race time. And the person had here an example with numbers and would like to know if is correct the calculation. I don't know if you want to answer now, or you want to analyze this better and then the person contact you. We had already put your email here.
I have read it actually, and I've forgotten now a tiny bit, but I when I looked at it, the answer to this is yes, you're right. This is how to do it.
So
yeah. It's very simple.
A simple.
And actually, I think it's also quite evident from the calculations that I just showed on my slides. But I think this is quite right. I'll have another look at it, and if I find any errors, then I can simply convey that to you, and you can send it to the person who submitted. Yeah, okay. I would be very, very happy. Sorry.
The person is me, Gunnar. Thank you.
Hello, Vanessa. Hi, it's good to see you. And congratulations with your new president.
Okay, yeah, this was my doubt, because I'm trying to know if I really understood the video and also the...
But I think you do understand it quite well.
Okay, and now I feel more comfortable. Thank you.
Yeah, it's great, great. And I will definitely contact you if I find that there is an error in it, but I don't think it, I think it's quite correct.
Okay, thank you.
Yeah.
So we have another one, how to control variations in electrophoresis that do not impact the percentage tail DNA analysis. Are there data showing how much can be varied in the time of unwinding and electrophoresis, and that it does not impact analysis between different tests? In visual analysis, this difference ends up not impacting the result of different tests, since the damage class is considered, and that these do not vary in the winding, unwinding, or electrophoresis time varies a little.
I may be a bit subjective in this, but and also I don't know, of course I don't know all the data here, but in my head and my understanding, then the unwinding hardly affects the outcome. Actually, you may vary the unwinding time quite a bit, but the electrophoresis is understood, and and mechanistically understood and should be corrected for, and that is actually quite important. There are other stages in the Comet assay which might be important, and one of them is the lysis. And if you have extended lysis conditions, then that will lead to modifications of base damage, actually. And we have one paper on that when we used glucidomide, which is a metabolite of acrylamide, and that leads to guanine residues. And if you treat cells with acrylamide, no, with glucidomide, sorry, and you lyse overnight, and then you use the FPG enzyme to detect these residues on the guanine caused by glucidomide, then these residues actually slowly destabilize the guanine, so that the FPG enzyme then can attack the lesion. And if you lyse for just 1 hour, then you would see much less damage compared to if you lyse overnight. So that's simply one example of a mechanistically fairly well understood situation in which you have a special type of DNA damage, base damage, and then you do have a significant effect of the lysine conditions. So be aware of that. If you are working with unknown chemicals and you study and you didn't know the mechanism or the way of action or something, then you should actually be aware of that, and you might try to vary your lysis conditions. You might try to vary the pH. You can also try to vary its duration. That means either very short or very, very long.
Okay. And we have a final question, at least that one that we receive. What protocol for electrophoresis would you recommend for sperm cells Comet assay, namely the duration of DNA unwinding time and voltage centimeter in alkaline buffer?
Yeah, this is a very extensive and quite an interesting and a bit difficult issue. We have been, myself in my group, we did a lot of work with sperm Comet assay, and we made a review on that together with, I think it was Gordon, wasn't it, a while ago, and there is a lot of data there. I think the person who posed that question might look up those papers and read them. There are several protocols for preparing the naked sperm for sperm Comet assay, and these protocols, well, this might be debatable what I'm saying now, but some people might claim that their method is the best, and I'm not so sure that there is actually a winner here. I think you might, it's difficult in any case to do the sperm Comet assay, and that has to do with the chromatin, how the chromatin is organized in human sperm, which is very different from my sperm. That has to do with the proteins being structuring the DNA rather than the histamines. No, the histones, sorry. But it also says something here about electrophoresis conditions for the sperm Comet assay. I don't think that is the electrophoresis conditions themselves should be different for the sperm Comet assay, because once you have actually made nucleates and taken away, hopefully, most of the proteins covering the DNA, then the electrophoresis conditions should not be much different from what you do when you have the somatic cell DNA. During the years, we tried to find suitable standard treatment procedures for sperm, actually, both for human sperm and for mouse sperm. And that turned out to be very, very difficult. And the observation is that if you use ionizing radiation to damage sperm DNA, then you don't at all get the same rate of damage formation as you do with somatic cells. You must go up to let's say 10 to 50 times as much radiation dose. And this is very surprising, but it probably has to do with the way that the DNA is packaged, so that there is a bit more inaccessible to the effects of radiation in water, which are the hydrolysis products of water. That's one possibility. Another possibility is that it's difficult remove all the proteins around the DNA. So therefore, the strand breaks are not so easy to analyze in the Comet assay as they are in somatic cells. So this is very critical, actually. I would be happy to communicate directly with this person who posed this question.
Okay, we put your email already here, and we had another question that was made here in the chat by Eleonora. She said that she was wondering if you have any insight into possible interference of nanoparticles with any step of the assay.
Oh wow, that's quick answer is no. I don't know much about that. I know that there has been attempts to analyze nanoparticles with the Comet assay, but actually I'm afraid I do not know the result of that. I wish I could be more helpful.
Oh, okay.
But again, I do communicate with me. I will be happy to answer questions also when I get more time to think about them.
Thank you so much, Gunnar. I don't know if any person want more to make some questions. I wonder that no. I will thank you again, Professor Gunnar, to accepting this challenge.
You're welcome.
And thank you so much. And now I would like to pass to our second expert. So our second expert is Dr. CARLA COSTA from the Environmental Health Department of the National Institute of Health in Porto, Portugal. Dr. CARLA COSTA holds a PhD in biomedical science. She is currently assistant researcher in the Environmental Health Department of the National Institute of Health and leader of the Life Force Environmental Exposures Lab of the Laboratory of Integrative and Translational Research in Population Health. Over the last 18 years, she has developed a strong expertise in genotoxicity assessment, both in vitro and in population target studies. Also, she is involved in more than 10 projects as researcher and also as PI, and she is also invited professor in the School of Health of the Polytechnic Institute of Porto. So, Dr. CARLA, thank you so much for accepting this invitation also. You are already sharing your screen, and the title of this talk is the use of placental cells in Comet assay. So thank you so much for accepting this.
Well, good afternoon. Thank you for the invitation and the introduction. As you were saying, today I will talk a little bit about the use of placental cells in the Comet assay. Our group has been working in the Comet assay for the last 20 years in different matrices. And today I will tell you a little bit about the use of these assay with the placental cells extracted from the placenta. So this was a need that comes from our field of work. And we mainly work, our main field of work is actually to try to understand how different exposures can induce some kind of health problem. And we have been working in different, looking at different chemicals or different agents. So you have here, for example, chemicals or radiation, occupational risks, agricultural practices. So we have been working in some of these different fields, and our common line is the study of genotoxicity. So as I was saying, our main goal is to try to understand which is the outcome or the disease that comes from a certain type of exposure and a certain type of source. Of course, that it is very important to consider not only the source and the exposure, but also the timing of that exposure. Because we do know that depending on the time, on the life stage that we are at, the effects or the type of disease that we will find will be different. So this was why we decided to initiate a new line of research, trying to understand or explore which are the possible effects of chemical exposure during the fetal stage. This slide that I bring you here is a slide that I actually care a lot because it shows the interaction between the different disciplines and how important it is to have different experts working in the same project. So what you see here is actually the continuum between what people say, the continuum between exposure and disease. And you can see that we will have the traditional environmental science in the beginning, and then we need also to understand exposure, and for that we need exposure science. And then we have a stage at which we need to understand how the chemical or the agent interacts with the human body. So we need a lot of information about toxicology. And then we need also to have experts in epidemiology, because we need population studies. So in order to have information or actually to have relevant information about what are the possible effects of certain chemicals or of certain agents, we do need experts in different fields. And this is where I want to introduce you the human biomonitoring, because this is pretty much what we have been doing in all these years, is to make use of human biomonitoring to try to understand, on one hand, the exposure levels that the populations are exposed, and on the other hand, the effects that can be the result of those chemicals. So in the case of bird studies, and this is, if we can look at this in a broader perspective, so we need to have a research question. We need to know which is the chemical that is important and what do we want to study. And then we need to choose our population. In the case of bird studies, you will find studies that will look only at pregnant women. There are other studies that will look at mother and newborn pairs, and there are other cases that only look at newborns. So this will depend on your research question. And then you will need to choose your biomarker. So you will find lots of information about exposure biomarkers. And those biomarkers are really important to understand which chemicals are we exposed. Do we need to prioritize the study of their effects? And you also have the biomarkers of susceptibility. And if in the end you will have your results, and hopefully you will have some kind of conclusion. But in order to put this rolling, we need to choose also which biomatrices will we use. And this is actually very important in the case of bird studies. And this is because we are dealing with susceptible populations. So we will have some ethical and some practical limitations. And considering this, I would say that it would be much more easier if we can find matrices that are non-invasive or that can be collected in routine. So if during the usual medical appointments, women can provide those samples for the birth studies. I'm showing you here some of the most common matrices that are collected for these kind of studies. And you will see here for example, exhaled air, urine, and that is common, or hair, that is common to many other studies. But then we have also breast milk, for example, and then we have the placenta and the cord blood. If you look at the literature, you'll see that cord blood is probably what is considered the gold sample. And this is because it is much more related with the fetus. So we will have information, much more information about what is the possible exposure and what is the possible effect to the fetus. But at the same time, the cord blood is a very limited and very valuable sample. And in many cases and in many countries, it will be difficult to collect cord blood because we have the national public banks of cord blood. So it will be difficult to have this kind of sample. On the other hand, we have the placenta. And the placenta is often discarded. And if you talk with any doctor, he or she will tell you that there are cases in which the placenta is, of course, collected. And many times, if it is necessary to do some kind of histologic or any other kind of exam, but many times it is just discarded. However, it has been people have not been using that much the placenta because it has lots of different cell types. And this is normally considered to be a problem for some assays. So people try to, are not using the placenta as much as it could be. So a few years ago, when we initiated a study, and actually the first one, it was a collaboration, we looked at the literature trying to identify what could we find about the Comet assay and its use in placenta cells. And we actually could only find 2 studies. So one of them was this one, and it was about cell characterization. And as you see there on the right side, cells were isolated by enzymatic digestion because they were used for culture in a second stage. And the DNA stability was analyzed by the Comet assay. And then we found a second paper in which cells were also isolated by enzymatic digestion because they were also cultured. And because they were treated, they were isolated and then treated by cigarette smoke extract. So these were the only papers that we could find. So our first, and then based on this, and not much information, so we needed to decide how would we do it, and we had to decide different points. So I will tell you just a little bit about how we collect the placenta, how do we isolate the cells, about a little bit about the freezing of the cell suspension, some details on the Comet assay protocol, and then I will just answer you the most frequently asked question, because there are many groups that ask us this many times, if it is possible to obtain the cell suspension after cryopreserving the entire placenta or the entire piece. So considering the tissue collection, so I just, I wrote here just 2 sentences about some evidence that you can find in some papers. So the placenta tissue is actually very valuable because it is also mainly a fetal organ. And there are many studies showing that these placenta changes are directly related to fetal development. So placenta can actually be a very good proxy to the same kind of effects or the same kind of alterations that we would find in the cord blood or in some fetus tissue or matrix. So and at the same time, it has also been proved that genotoxic compounds have the potential to induce genomic damage in the different cell types of the placenta. And I'm showing you here some the indication of the paper so that you can later on go to these papers and take a look at this information more carefully. So what we did was asking to the nurses in the different studies to collect a section of the placenta. So they should place the placenta in the bench looking at the fetal side, so you can see the umbilical cord insertion, and then they would collect a piece at least 4 to 5 centimeters away from the insertion of the cord. So in the end, we would have something like this. So these were the recommendations. So collect a tissue section in the fetal side, at least 4 centimeters from the insertion, and then we would ask them to put this in some physiological solution and kept in the fridge until further processing. Let me tell you that this further processing was quite quick, because we were not having the samples for many hours in the refrigerator. So most of them would be in the maximum 8 hours in the fridge until coming to the lab. Okay. Then we need to think a little bit about the cell isolation. Would we be doing mechanical separation or enzymatic digestion? As I was showing you before, the 2 papers that were available were indicating enzymatic digestion, but they were doing culture. They were culturing the cells after that, and we did not need that. So we decided to test the 2 different protocols. And for the mechanical separation, we used a very standard protocol, just using scissors and then going through a syringe. So it's a quite simple protocol. And then we did also the enzymatic digestion. And this was based on at the time we were working with the medical university, and one of the colleagues had done an internship in the Yale University, so she had this protocol, and we adapted that protocol that she had to do this enzymatic digestion. She was using trypsin and DNase 1, so we adapted the protocol. The paper that I'm indicating here is not the actual protocol, but it's one that is publishing a very, very similar the steps, very, very similar steps. So you can have an idea of what would be the protocol. And then we just compared. And you can see here the results of the blue bars are the standard protocol, and the mechanical separation, and the red bars are the enzymatic digestion. So in the end, what we could see in the microscope is that actually the mechanical separation would provide a slightly lower levels of DNA damage, and it was much more simple to carry out. So we decided just to move forward with the mechanical separation protocol. So you have here very simply what we were doing. We were washing the placenta piece using a cold PBS. Of course, this was all done in a cold plate. Then we were adding the PBS and mincing the tissue using scissors. Then we were recovering 2 milliliters of cell suspension, and we were avoiding any larger bits of tissue, and we were running it very slowly through a needle. And then we were washing this twice, again in PBS and at a relatively low speed. So because we were collecting like 1 placenta today, 2 placentas tomorrow, it was not feasible to do immediately the Comet assay. So we were freezing all the samples after the manipulation. After obtaining the cell suspension, we were freezing the cell suspension, and we were freezing them just with 10% DMSO. Of course, we were doing lots of different aliquots so that we have a reasonable number of cells in each aliquot. And then at the time we want to run the Comet assay, we just need to centrifuge a volume that contains approximately 25,000 cells. And then we just add, remove the supernatant, add low melting point agarose to the pellet, and then we use the 12 gel system. So we just do 5 microliter gels. And we use the usual solution, so I'm not providing that information here, but we do the lysis just for 1 hour, the alkaline treatment for 40 minutes, and electrophoresis for 20 minutes and 1 volt per centimeter. And now looking at the final question that I was talking about, can we do the cell suspension after the placenta cryopreservation? Well, we tried, especially because we had another group that was very interested in doing the Comet assay, and they had the placentas already frozen. So they wanted to isolate the cells after doing the entire piece preservation. We tried, but we couldn't. The DNA damage levels were much higher. So in the end, we just gave up, and we couldn't do that. So this is our experience. Of course, you may find, and we try to replicate, there are a few papers talking about isolating cells from frozen tissues, but we couldn't do that. But anyway, you can always try. There are some papers describing those protocols. And this is it. This was what I had to show you. I hope it was useful to you, and I'm now taking any questions you may have. I know that there are some previous questions, so.
Thank you so much, Carla. It was very useful. Yes, your presentation is a type of matrix that has been raising a lot of interest. And as you say, and well, we have already some questions that were added in the Google Forms. And I will do the same as I did in the GUNNARs. That is, I will put in the chat also the question. So the first one that I want to tell you is, washing the placenta is washing the placenta just sufficient to remove blood cells?
Well, yes, you'll see that in the beginning, we'll start with a very red solution. The cell suspension is really red, but as we do the washes, we will see that the cell suspension becomes clearer and clearer. Of course, it will have some red blood cells. In our case, because we freeze the samples, usually when we thaw them, in the end, we don't get the red blood cells, or pretty much none of them. But in any case, even when we do it fresh, they won't interfere with the assay. So it's I think it's okay to just to do the washes. You don't need to have any further steps.
So the second question is, how can mechanical injury be prevented during tissue homogenization?
Well, this is a very good question, because in fact, using the needle often increases damages the cells. So if you see that is a problem to you, we don't see that. We see lots of damaged cells immediately if we don't do the washes. As we wash the cells, we will remove that debris, and the images in the microscope become much more clean. But if you still see some problems, I would say that you can probably try to use a cell strainer instead, to see if it probably the yield of the number of cells that you will collect will decrease, but maybe you will see cells that you won't see such injured cells.
Thank you. We have another one. Do you isolate specific subtypes of cells from placenta prior to Comet assay, for example, trophoblast, or do you use placenta-derived cells in general?
We don't isolate, but there are many protocols that that do that. Actually, some of the references that I was showing you in the slides today, they will have the protocol not only for the enzymatic digestion, but also for the isolation and purification of cells. If you want to have a certain type of cell, what we just did was to avoid, when we were cutting the pieces, we were just trying not to get the connective tissue, or if we see some bigger vessel, we were not using that. So we were just trying to get the spongy tissue, and that will help at least not to get other very different types of cells.
Okay. We have here also Jamila Rocha saying congratulations to you, Professora Doutora Carla Costa. I'm a great admirer of your work.
Oh, thank you.
Okay. And then we have another question that is not specifically focused in this work that you presented, but I will put also here because we still have some minutes. So, would it be possible to carry out an experimental design to evaluate genotoxicity of women who live in agriculture areas and are exposed to pesticides? Do I mean random collections or patients who agree to be part of the research? So it's more a human biomonitoring question that is your field also. So
yeah, well, I would say that if your purpose is to see if the levels that women are exposed to pesticides in that particular context, if you want to know if they are somehow damaging the placenta, well, I would say yes, you can do it. There are other studies doing that, not using the Comet assay, but doing other types of biomarkers in the placenta. What we need to make sure is that we are choosing a biomarker that is adequate to the effects of the chemical that we are analyzing. So in this particular case, you just need to make sure that the pesticides that those women are exposed to are actually genotoxic, just to make sure that you are choosing the right biomarker.
Of course. Questions from the Google Forms, I did all. I don't know if someone want to ask something else to Carla. I imagine, Carla, that the most difficult part is to access this kind of samples, because if we think in placentas, in the end of the birth, of course, all of them will be discarded, okay? But in the case, the work that you did, it was a medical doctor from obstetrics that was involved also in the research team. So he would collect from yes.
Well, we couldn't go to the birth room to collect the samples. So what we were doing was we were leaving a kit with all the necessary material for the collection, and we would leave them in the hospital every day. We were leaving these new bags with all the necessary material. Women were identified because they had been contacted before. So when they went to the hospital for delivery, they were identified. And as they went to the birth room, the nurses would get one of those bags, and they were immediately collecting all the samples. And so, yes, we needed to count with the cooperation of the nurses and also the physicians, because they were the ones in charge in that moment. And at the same time, when you read the papers, you will see that we recruited 840 women around that. And in the end, we only have samples for 600. And this is because in all the other cases, the medical staff found that the context or the situation was not perfect for sampling. Okay. So it was their choice not to get the samples at that moment. And this is something we need to respect, of course. This is their decisions. They are the ones there. And so, of course, we lost lots of samples with that, but it is really necessary that that kind of decision is made by the medical staff and not by us.
Of course.
Can I ask you a question?
Of course.
CARLA, thank you for a nice talk. Many years ago, I was a bit interested in placenta, and do you know anything about establishment of biobanks for placenta?
No, not that I'm aware of. Not in Portugal, for sure.
Yeah.
We don't have any.
We tried in Norway to sample placenta from various parts of Norway and freeze them. And the idea was that then we should have a very good tissue bank for searching for and studying possible effects on placental cells in the future. Because I mean, you can get so much tissue, and it should be possible actually to store it fairly cheaply. And then if we run into unexpected environmental problems in the future, unknown new synthetic chemicals or something, and high exposures, then we could actually go back and collect it. But we didn't get financing for it. But you have not heard about anything being done in other countries? No.
No. What I see is that placenta is being increasingly used because there are lots of biomarkers that you can do using the placenta. And at the same time, especially if you want to do biomarkers of exposure, you can also try to use the placenta instead of using the cord blood. That was the sample that was previously being used. So there are lots of things that can be done. And the scientific community, I think it's much more aware of the possibility of using the placenta instead of using the cord blood.
It should also be possible to, or it should be easier. I mean, it should be easier to get permission both from the owner of the placenta, from the donor, and also from the medical community, wouldn't it, to do this type of research? I think it would be quite valuable, actually.
Yes, it's quite easy. I mean, most of the women are willing to provide the placenta for research, unless and that is not the case for cord blood, because in the first place there are the private banks for cord blood, and some of them are already had already bought that service. So in those cases they could not be enrolled in these studies, and even if they haven't bought the service to a private one, it was also a priority to collect for the public bank. Because we have the public bank of cord blood. So it was actually just the little bit in the end, because they collect a certain amount of volume, and then a little bit in the end, if there was still cord blood, then it could be collected for the project. But that is just for the cord blood. I haven't heard anything about the placentas.
Yeah. Many years ago, I was involved with New Generis, and many others of you I know were also involved. And we collected a lot of cord blood in that project, that EU project. It was FP6 or something, I believe. But that was very tricky, and it was difficult to get ethical permission and to store it and everything. And also it's very limited in volume, as you know. So I would really go for placenta.
Yeah.
We have three questions in the chat that we're adding. So the first one from Sabine is, does the time of collection after birth of the placenta influence the DNA damage?
It is a possibility. We have that kind of information. We have the time of birth and
of
I'm just
collection?
no, I'm just thinking. I'm not sure if the question is about collection after birth or processing after birth, because collection after birth, we were asking the nurses, and the nurses were doing that immediately. I mean, that was done by one of the nurses that was involved in the birth, but of course there are people that are taking care of the mother, and then there was another nurse that was doing that immediately after the birth. So it was very quick. What we are trying to see, and we will have that when we have our entire samples analyzed, is if the timing between getting the piece from the hospital to the lab and processing it, if that can actually influence the DNA damage. Because we do have that information. We know the time of birth, and we know at what time we collected the samples in the hospital. So we have the information, and we'll see that.
Okay, another one is, do you think it's possible to adjust for the red placenta?
I would try. I think this can be a starting point, and but I would say yes. But of course, you will need to try. But if you have any questions, I'll be happy to help.
Okay. And another one, have you compared in your studies the difference in DNA damage between placenta and different cells in the mother's body?
That is one of the aim of our projects. Yes, it is to compare. We collected peripheral blood from the mother, the placenta, and the cord blood. So one of the aims is actually to compare and see how the DNA damage in these 3 different matrices will relate. So it's one of our aims, yes.
Another question here. We noticed some effects on DNA repair in relation to time of collection after birth. So is the question from Sabine. That is why I ask, good to keep in mind indeed the processing time, and thanks for the presentation. Okay.
Thank you.
Let's say in DNA repair. Carla, I add your email here because there are persons that would like to know if could have your email to make some questions about the project. I hope it's not a problem.
No, please go ahead.
I don't know if someone else want to make some question more. So in that work that you are doing, you are expecting to make public when?
It's a good question. So we started with the questionnaire analysis. So we have a paper now last year about the socioeconomic status and exposures. Then we were validating the questionnaire against a biomarker of exposure. So it was cotinine, and that was also already published. And now we are working in the samples, but of course there are hundreds of them.
Yes.
And it will take some time.
Yes, many time and a lot of work, because if you will search in different cell matrices, it will be huge work and very add value. When I'm curious to know about the relations and probable correlations that you will have
between different matrices. Yes.
Yes. So if nobody have any more questions, I would like to show again my appreciation to both experts, so Dr. Gunnar and Dr. CARLA, for accepting this challenge and joining here with us and the future researchers and all researchers, because there are still questions and doubts regarding what we are doing in Comet Assay. So thank you so much to you. Thank you for all the questions and the participation. I hope it was fruitful. Any question or doubt that comes, you can also email the experts. The emails were already presented here in the chat. And again, remember that this meeting can be revisited on YouTube in the ICAWG website. I wish you all good experiments and thank you so much for having you here. Thank you, Gunnar and Carla.
Thank you.
Okay.